Good afternoon and welcome to the Kirby seminar program. Welcome everybody. Today we're going to hear about some backroom work that was done as a critical part of implementing point of care testing for COVID-19. Before we start, I'd like to acknowledge the traditional owners of the lands on which we all gather today and pay my respects to elders past, present and emerging, and extend that respect to Aboriginal and Torres Strait Islanders who may be joining us today. Again, welcome everybody. To just clear some housekeeping before we start, there will be a presentation today, will be given by both Steve Badman and David van Bockel, so a little bit different from the normal format, and then after both have presented, we will open it up for questions. To ask a question, the simplest way to do that is to go to the icon on the screen with the question mark in the middle of it, click on that, and then you can enter your question by writing that in. Please put your name associated with that so we can say who the question is from, and it is possible to upvote any question that you think is most interesting by clicking on the question and supporting it. In terms of our presenters today, we have two, as I said. The first is Steve Badman, who's spent the last 10 years working on the implementation of point of care testing for viral and sexually transmitted infections, particularly in resource poor settings. And he has worked in a range of international venues, including Vanuatu, Fiji, PNG, Myanmar, Thailand, Indonesia, Malaysia, and also with remote communities in this country. He has a diverse and specialised skill set, including point of care implementation in remote and middle income locations. Our second presenter today will be David van Bockel, who has a long background in defining assays to look at the correlates of immunopathogenesis in a range of viral infections, particularly HIV and HPV, and has conducted these assays in the context of both cohort studies and a range of clinical trials. So I'll now hand over to Steve to open the presentation. Thanks, Steve.
Thank you, Tony. Just before I start, I wanted to thank all of the authors that you can see listed on the first page of our presentation today, and also extend that thanks to the many team members in the Aboriginal and Torres Strait Islander COVID-19 point of care testing program for their contributions to this presentation today as well. I'd also like to acknowledge the significant cooperation of Indigenous health services, state health services, jurisdictional bodies, laboratory networks, and of course the Commonwealth Department of Health. I'd also like to note that there are no conflicts of interest to declare, and that all the expert tests used in the studies described in this presentation today were paid for by the Aboriginal and Torres Strait Islander COVID-19 point of care testing program. So the story of this evaluation really begins in March of 2020, and for many would be familiar with the city of Wuhan in China, where they are reporting increasing numbers of COVID-19 cases, and the rest of the world is increasingly realising that the pandemic will not be confined to just this part of the world. At a personal level, I realised this on March the 17th at the Brisbane airport when our Prime Minister suddenly announced there would be a closure of Norfolk Island, which is an Australian territory off our east coast. And suddenly I get bumped off this flight at the boarding gate just before, minutes before the departure. In the background, many other emergency measures are underway. And as a part of this, the Commonwealth Department of Health seeks to expand COVID-19 testing across remote Australia and looks to the Kirby Institute and the International Centre for Point of Care Testing at Flinders University given 31 remote Indigenous health services are already established within the Tango 2 program, which is an extended program for STI testing in remote settings, and all of which use molecular GeneXpert testing at the point of care. So, based on the extensive planning and implementation of that Tango 2 program, a COVID-19 remote testing operational model is developed to inform and guide this new planning and implementation process. By this stage, the number of COVID-19 tests available for purchase in Australia has become clearer, so a test distribution model and a supply chain pathway is also established. At the same time, state-based COVID-19 program coordinators are conducting extensive site and risk assessments and enrolments from health services interested in becoming a new COVID-19 testing site. In parallel, other team members developed a detailed quality assurance framework, and we manufactured approximately 3,200 in-house positive and negative COVID-19 quality control swabs to be used for GeneXpert instrument verification on arrival at sites and for the running of monthly QC within this particular program. COVID-19 GeneXpert training across these sites is simultaneously provided by a very skilled team at Flinders University, using remote access only, I might add, and this has resulted in some 230 competent COVID-19 expert operators being certified. And as you can imagine, many other technical and practical aspects were being rolled out at the same time. And just like that, 5 months later, the implementation of COVID-19 testing at all planned sites is completed. Suffice to say, this has been a massive undertaking, and we believe it's the largest single implementation of GeneXpert testing in the world, with 86 sites, testing sites now operational across Australia in all states and territories, except for the Australian Capital Territory. And just to give you some idea of what that rollout looks like, this is a map of testing coverage and shows many of the 86 sites included in the current program, and as far up as Boigu Island, which is just off the PNG coast. And I'm just going to activate my pointer here just to show you how extensive the coverage is in relation to COVID-19 remote testing as of the end of September. In the second cutout here, this shows Central Australia and gives you a better idea of how the hub and spoke model we are using is being used to cover or expand testing coverage. In this case, the point of care instrument is located at a hub site, which is signified by a round dot with active on-site testing. An outreach sample collection is conducted at sites with those symbolised by a triangle, and the samples are returned to the hub sites for testing. So how did all this happen? Well, just to give you a visual overview of the operational model used within the Aboriginal and Torres Strait Islander COVID-19 point of care testing program, this model starts with stakeholder engagement and works its way downwards in a stepwise fashion to establish and operationalise each component that you see on your screen. What is not visible, of course, is the amount of adjustment and fine-tuning within and across components as the model rolls out and beds down over time. What is important to know, however, is that this model is very consistent with the Australian National Point of Care Testing Guidelines and complies very strictly with those particular requirements. So it's obviously a process that was unfolding very quickly, and in the early stages of planning, it wasn't clear to us how many COVID-19 expert tests we would be able to buy per month, and if we were able to match viral transport swab kits to the number of tests needed to for us to be able to supply those to COVID-19 testing sites. So an international search began on our part to see if an alternative VTM swab kit supply or supplies could be secured. And initially that search revealed a product made in China which claimed to deactivate the COVID-19 virus. Based on this information, discussions began to unfold about the potential benefits of this type of media if used for COVID-19 testing here in Australia, and the need to validate its effectiveness in killing the virus. So to avoid any shortfall in viral transport media swab kits, we imported some of the China-made viral transport media tubes and swabs, and you can see that this is an image of that product in the top of your right-hand screen. At the same time, as a part of our due diligence, we started exploring other FDA registrations and discovered another product called Primestore MTM, which has also had a claim to deactivate some viruses, but not COVID-19. Further investigation revealed this product was first manufactured in 2016 and developed originally to deactivate H5N1 virus samples. We then found further validations and publications which confirmed MTM's ability to deactivate flu A/B and also tuberculosis. And in the bottom of the slide here, you can see a product which is known as COPAN eNAT. And this particular sample transport medium also came to our attention, given it had a viral deactivation claim, but it was a more recent entry onto the market and did not at that time have the same depth of performance evidence as the Primestore MTM and could not be bought in Australia. And that remains the case today, as far as I know. So with the cooperation from colleagues in Western Australia and Sydney, we scrambled and we were able to pull together enough Primestore MTM and eNAT vials to undertake a transport media performance evaluation at the Kirby PC3 lab. And as you can see, from these images, the study took place under very controlled conditions. And I believe in this bottom image here, that's Associate Professor Stuart Turtle conducting a review of the SARS-CoV virus under a microscope. In June 2020, the transport evaluation was completed and a manuscript has since been developed and submitted for publication. And I'd now like to invite David van Bockel to give us an overview of the methods that were used for this evaluation and the primary results that came from this work. So I'll hand this over to you, David.
Thank you, Steve. So as Steve has mentioned already, we decided to test several types of reagent, and you'll see them listed in the lower left-hand corner of the slide. There were 3 types that were thought to be compatible with the main GeneXpert system, which contains a principal inactivating ingredient known as guanidine thiocyanate. These media were the Primestore Molecular Transport Media for nucleic acid testing, media from the company COPAN, the eNAT, and another universal transport medium that Steve's already mentioned that I will refer to in this slide as CV19, which was imported from China. And we also included our own viral inactivation buffer from the Kirby company to ensure that the inactivation process was successful. Medium that was meant to support the virus and keep it alive was also tested from the COPAN company. And furthermore, we're interested in testing inactivation of SARS-CoV-2 using a disinfectant, Sterogen, and using heat as an alternative to the commonly used liquid bleach as a cleaning agent, which is generally incompatible with guanidine thiocyanate. We tested the media provided by Rebecca's team in 2 ways. Both methods started in the same way by the addition of pure SARS-CoV-2 virions at a concentration that we already knew. Directly to the liquid from the collection tubes. We either added the mixture directly to the cells, which we knew could be infected, and these are called Viro E6 cells. You can see them attached to a dish in a picture in the top right-hand corner of the screen. We first used a method dubbed the WHO method, or the routinely used World Health Organization method for detecting virucidal activity of chemicals. This method was chosen to determine the minimum amount of time we would need to inactivate the virus. We were able to add the mixture and then dilute in cell culture media to a point where we could see the infection of cells again. This is at a minimum of 2 minutes. However, given the media can be highly toxic to cells at high concentrations, we used another method to test for sensitivity. We called it the BDX, or buffer exchange method, which was designed to keep the cells alive at concentrations we knew were already high by removing the inactivating agent using columns that can be spun in a centrifuge, washed, and then added back to the cells to see if there was any virus remaining. The cells were infected for 5 days. And then to make sure the infection took place, we reinfected fresh cells with media from the first plate. At both times, we looked for evidence of cell death. You'll see that in the bottom of the screen. These are called plaques, or I'll refer to them later as a cytopathic effect under the microscope. And also by picking up viral material in the media using the same molecular test used to detect SARS-CoV-2 on flox swabs that were performed in collaboration with the virology team at Prince of Wales Hospital. Next slide, please, Steve. So in this slide, I'm going to show you an example of how we test the effectiveness of these media in deactivating SARS-CoV-2 by measuring the number of cells remaining at the end of the incubation time. The cells we stain with a dye that make the nucleus glow with a blue color when excited with a particular light. That glow or fluorescence can be counted using software after a picture has been taken of the cells using a microscope. Over the titration curve that I showed you in the previous slide, you can see a halfway point where the virus is inactivated, and that is an important unit of measurement for virologists. On the left side, images you see under the microscope. You can see that for Primestore, COPAN, eNAT, and QIAGEN, AVL, where the reagent has been removed by washing using the spin columns, you have a healthy lawn of cells. To the right are the control wells, where virus was passed either directly over the column, washed, and then plated out onto the Viro E6 permissive cells. You can see that the lawn begins to look a bit threadbare in the picture at the top. This is the effect of the virus, which is killing the cells, and the nucleus can then no longer be counted. The picture below is a virus added straight to the plate and is the assay control. On the right, you see the resulting titration curves for inactivating virus using example nuclear counts at either a low count in the column, at a low count of virions, on the column to the left, or a high virion count in the column to the right. In both cases, the positive control titration curves that you can see in red worked very well to kill Viro E6 cells that required a significant dilution of the virus going from right to left on the graph before the cell count starts to return to normal. The first test method, the WHO method, was quite toxic to cells at high concentrations, and that meant that cells were effectively lysed instead of killing the virus in a uniform way, as you can see in the top right-hand graph. That's shown in pink and blue, This is before the cell started to recover at about a 1 in 1,000 dilution of the reagent. Importantly, we did not see any cytopathic effect from the virus detected after cells started to recover from that cytotoxicity, meaning that the virus was inactivated. To confirm this though, at high concentrations of SARS-CoV-2, using the buffer exchange method, cells remain viable from a high to a low dose of SARS-CoV-2, as you can see by the flat curves in the two middle graphs. This was also the case for inactivation of the virus using Sterigene disinfectant, as well as the more traditional applications of heat, seen in the graph at the bottom in green and gold. Next slide, please, Steve. So this table demonstrates the test results in full by measurement of cell count at the most appropriate point, halfway along the titration curve. They include the PCR test results at the same time point. And what this table will tell you is the effectiveness of the different reagents in inactivating SARS-CoV-2 using multiple detection methods. So results that are positive for SARS-CoV-2 are shown in red, and results that are negative for SARS-CoV-2 are shown in black. On the left is the WHO method, the table on the left-hand side. which is measuring the presence after the cells have recovered from the toxic effect of the reagents, as I've already mentioned. The assay controls were positive for SARS-CoV-2 by a lowered cell count in the top 2 rows, showing evidence of plaques and detection of virus by PCR. There was no virus present in the toxicity controls in the rows below, which contained only media. This was the same for Primestore, COPAN eNAT, and the virolysis buffer control. The same was observed when we spiked these media with the virus that were added in parallel. Interestingly, the COPAN and CV19 China VTM media that was considered to be an inactivating medium for both, they both tested positive for SARS with a lower cell count, and they both detected positive for the virus using molecular techniques. These results were then repeated in a table on the right-hand side using the buffer exchange method. The negative and positive controls for the assay were respectively negative and positive for the virus, as determined by the presence of high and low cell count, as well as the detection of viral products by PCR. All molecular testing media using the buffer exchange method were negative. That includes Primestore, the AVL, and the COPAN eNAT, indicating that we were able to inactivate SARS-CoV-2 using these media at the highest concentration of virus that we had available in the testing facility. Again, the COPAN support medium and the CV9 inactivation medium were both positive when we measured by cell count, and were positive for SARS-CoV-2 viral products that were detected by PCR. We queried the CV19 product on these results with the original company that provided the media, and which advertised that they did contain guanidine thiocyanate, and the company was approached for comment. We also measured for the presence of guanidine salts using time of flight mass spectrometry, and detected these salts at what appeared to be a significantly decreased amount in this media compared to those others which are commercially available that we had tested using the same program, specifically Primestore and COPAN eNAT. Next slide, Steve, please. So just to summarize, the protocols which we had previously validated, they inactivate SARS-CoV-2 after incubation in the viral transport media containing sufficient guanidine thiocyanate for 2 minutes, using the Sterigene reagent, and incubation at 65 degrees for 1 hour. We were also able to inactivate SARS-CoV-2 at a higher viral load with viable cells that were able to be infected. So these results were confirmed by multiple reportables via cytopathic effect, that is cell death, or detection of viral replication in culture. And I'll finish there, and I'll hand back over to Steve, who will continue the talk. Thanks very much.
Okay, thank you. Sorry for that small technical glitch. A little bit challenging operating from different locations and different people. So thank you, David, for a great summary of those methods and results in relation to each of the transport medias that were evaluated. In addition to those particular findings, we also wanted to establish if Primestore MTM was compatible with the GeneXpert platform. So we compared its performance against the Chinese manufactured viral transport medium and the standard COPAN viral transport medium, which had no viral deactivation capacity at all. Each media type was subject to a series of dilutions, making it harder and harder to detect the control material, and the results showed equal detection performance by all 3 media types when tested on the GeneXpert platform. In addition to that, another study is about to commence, so we can determine if COVID-19 samples stored in the transport media can remain stable over different time periods and at different temperatures. The purpose is to identify the stability of samples at these intervals, and this information will provide additional confidence when using Primestore molecular transport media in hotter climates and where immediate shipment may not be possible. As you can see from the laboratory evidence that David's just provided, Primestore MTM produced or an almost immediate deactivation of SARS-CoV-2 virus on sample collection, and offsets any risk when samples are transported or handled prior to testing. This finding also means that safety cabinets are not needed to handle or process samples when stored in this transport media, which is a major benefit, and especially where testing is conducted outside of laboratory settings or in settings where cabinets are scarce or unavailable. In terms of versatility, the Primestore MTM manufacturer's website also lists a number of known laboratory platforms this product has been successfully used on to date, such as Roche, Abbott, and a number of others. And also the published literature reflects similar evidence. So in the background of this lab work, the COVID-19 program continued to roll out, and the standard COPAN VTM was supplied to testing sites initially and until the viral deactivation study was finished. On receiving these study results, we immediately sought registration from the Therapeutic and Goods Administration here in Australia of Primestore MTM. Quickly flew in a 20-litre container from the United States, given it is not classified as a dangerous good. And with the help of Philip Cunningham's laboratory team at St Vincent's Hospital in Sydney, 10,000 tubes of Primestore MTM were decanted into 10ml tubes and labelled by his staff as quickly as possible. And I'd like to just take this moment to offer Philip and his staff a huge thanks, given that they did this large task in addition to their existing laboratory workloads. It was an absolutely outstanding effort. So the completion of this task enabled the MTM to be shipped to all 86 remote sites, and ongoing MTM shipments are now processed using an automated dispensing system in Brisbane and then shipped back to Sydney for redistribution to the existing 86 sites scattered across Australia. So before we shipped the MTM though, we informed sites about the change in product and added an additional guidance sheet into each pack of molecular transport medium tubes with information about spill management, given, as David's already mentioned, this product contains guanidine and thiocyanate. And why it's important to flag this product is it can have a minor reaction with bleach, especially if bleach is used to clean up, for example, a 2ml tube spill. This method simply requires that 2ml of MTM to be soaked up in paper towel instead, and the area cleaned with 70% alcohol to complete that procedure. Or in the future, Sterigene may also become a suitable replacement for bleach, and that could be used in conjunction with the 70% alcohol I've just mentioned. The other important point to note is that we changed the tube cap colour of the MTM tube to purple to make it easy for people to tell which was the MTM media and which was the standard VTM tube, given most VTM tubes on the market come with a red cap. So we wanted to be able to make sure there was no confusion about which product was which. And of course, then there's the often frequently asked question, which is, well, what does the MTM cost? And as you can see on your screen, the approximate cost at this point in time, based on small buying power and numbers, is around about $7.65 per swab kit. But when you consider that a standard viral transport media kit, which has no viral deactivating capacity, can sell for anywhere between $5 and $6 here in Australia. So I'd like to close today by simply saying that Primestore Molecular Transport Media with scientific validation may also be suitable for use with other infectious pathogens, which can transfer that particular infectiousness by aerosol or droplet contact. And this may be of value to both humans and animals. And to give you an example, here in the state of Queensland, veterinary surgeons are extremely nervous about taking samples from horses which are suspected of contracting Hendra virus. This is a virus that is transferred by bats through their feces, and the level of infectiousness and exposure risk to to those handling those samples is extremely high. While it may not be a product that necessarily gets used in the vector-borne industry, I thought it was kind of timely to perhaps just add this small cartoon, which suggests that in the future it may have a place in dealing with infectious pathogens that could be transferred by mosquitoes. And finally, this particular reagent could help in low resource settings where medical consumables, including PPE, may be in short supply. And to that end, we are talking with our partners at the Department of Foreign Affairs and Trade and at the World Health Organization to see if making this molecular transport media available in Papua New Guinea and the Pacific region is possible. And on that final note, I'd just like to say thank you and hand it back to you, Tony.
Thanks, Steve, and thanks, David. So we have some questions. The first one is from Philip Keane, who asks the question, why is it important to inactivate SARS-CoV-2 in the transport medium? I think this is really critical to the understanding of this and the OH&S issues that really dog working with SARS-CoV-2 in healthcare settings. So, Steve, would you like to reiterate why this is such an important critical step?
The key issue around why it's important is that once the sample is collected and it's placed into a standard transport media, if there is any kind of droplets on and around that sample, Philip, and or when that particular sample transport tube is opened either at the laboratory end or at the point of care for testing, there is an increased risk to individuals who are either handling or having to pipette active samples from that particular tube. Of course, the operator doesn't know at that point in time if the sample is active for SARS-CoV-2 or not. So being able to eliminate the presence of SARS-CoV-2 almost immediately on inserting the sample collection tube provides a major benefit and reduces risk significantly for those particular individuals who are either handling or having to process the sample at the testing end.
Thanks, Steve. So we might go back to that question to David. And it sort of follows on from that, I think, quite nicely. So just to remind the audience, what was asked was, why is it important? So you've gone to all this trouble to differentiate between the cytocidal, the cell killing effect versus the virus killing effect. And why is that important?
Thanks very much, Tony. In terms of the methodological approach, one of the most important factors to inactivation of virus is time. So it was critical to understand the amount of time required to inactivate the virus, and we needed to approach the methodology that was required to actually kill the cells while we're trying to measure the virus at the same time, because unfortunately in the laboratory we weren't able to use any other method to determine that the virus could be inactivated so quickly using the reagents in question. Now, in terms of the differential between virucidal activity versus cytocidal, we actually didn't understand before we commenced the experiments at what point we would start to recover cell growth. The chemicals are made of a vast variety of different components, of which we didn't know how they would impact on cell growth. So once we understood that they had a cytocidal effect in a very uniform way, we could take a very clear defining line between where we saw live virus from our positive controls in the experiments that were able to kill off these cells quite well, and the gap on that titration curve between the cytocidal activity demonstrating that there were logs of magnitude in reduction of virus as a result of incubating them in the reagents. So there was a great deal of so these products were very effective at killing the virus, and the cytocidal effect, I think, demonstrated that quite clearly.
Thanks, David. So, and Steve, if I can come back to you. So what is these tubes are going to be sent out to remote sites. How long can they sit there? What's the shelf life, and how often are you going to need to ship these tubes out to the remote sites?
Yeah, that's a very good question. Very good question, Tony. Actually, one of the points that I didn't include in the slide, just in the interest of brevity, was the fact that Primestore Molecular Transport Medium actually has a shelf life of 2 years compared to an average shelf life of 1 year for most non-deactivating viral transport mediums. And shipping is basically worked out on a very complex model, which is constantly revisited by the program every week based on the number of expert tests that are available to us. And they are obviously in very limited supply. So we monitor exactly where they're shipped to and how they're shipped and how frequently they're shipped using this particular model, but also based on the size of the population that a particular hub site may be servicing, and or the number of spoke sites that they have expanded sample collection taking place within that particular area, like I showed in the slide demonstrating Central Australia as one example. But obviously all of that sits in a very fluid position, because obviously if there's any change in testing rates, which are monitored through a centralised dashboard that actually looks at the number of tests completed per week by every one of those 86 sites. So there's a very sophisticated system of monitoring that takes place that determines when, where and how often we actually resupply those locations. Now, in 2 places in the Northern Territory, we send tests and consumables to a centralised location, and they distribute to sites in their area or jurisdiction based on a communication link between them and the health services that sit in their area. But for most other sites, it is based around the very close monitoring of that model.
Thanks, Steve. David, at the possibility of opening a hornet's nest, would you like to speculate on why the transport media that didn't work, or the inactivation media that didn't work, why it didn't work? Do you think it's down to the concentration of the guanidinium isocyanate, or do you think there were other issues there?
Thanks, Tony, and I'll keep my response brief. I feel that guanidinium thiocyanate is a very strong inactivating or reducing agent for viruses, and I strongly believe that any insufficient quantities of that in an inactivation media could be the main culprit in this case. And I think that given we took some extra steps to determine the level of guanidinium thiocyanate using mass spectrometry in these samples, and there was a clear difference between that and what was able to inactivate virus, I think we may find an answer in that space.
And Steve, if I can come back to you, so this is a little outside the topic of this talk, but perhaps the audience would like to hear how these 86 sites were chosen. What was the process by which these were chosen against other possible sites?
Yeah, thanks, Tony. It is slightly outside, but I think it's probably best to describe the fact that given that this program is led by Professor Rebecca Guy at Kirby and Professor Mark Shepherd at the International Centre for Point of Care Testing at Flinders, there is already an established program called Tango 2 across 31 of these sites in remote Australia, where molecular point of care testing is still taking place at the point of care for sexually transmitted infections in those locations. It then became a matter of discussion between the Commonwealth, the heads of this particular program, and also strategic locations in terms of other sites that might need to be chosen in regards to, you know, creating, I suppose, a defensive position, if you like, in regards to which locations might be better to be able to respond to and or manage not only having molecular point of care testing present, but also geographically well positioned to make sure that they'd be able to respond as well. So I think it's a combination of decisions that and information that was brought together, and also the willingness of new sites, bearing in mind that if there are now 86 or slightly over sites participating in the program, there were 31 in the Tango 2 program. That gives you, 45 new sites, and a part of that also came down to the willingness of new health services being willing to participate or feel that they were in a position to manage this kind of testing as well. So yeah, I suppose it's there's a little bit of a matrix, decision-making matrix there that was that brought all those key factors together.
Thanks, Steve. So if there are no more questions, we might wrap it up there. So thanks, Steve and David, for a terrific presentation. And I think what this demonstrates above all is the incredible collaborative effort on the part of really multiple people in multiple venues associated with multiple institutes who've really dropped everything else and pulled together to make this happen. And that you really need to be safe, attention to detail. And so I think what this, we've gone to extraordinary lengths to ensure the occupational health and safety of the operators in these remote communities, which were really important. And clearly, first-line healthcare workers are in the firing line with this infection, as has been repeatedly demonstrated throughout the world. And obviously, if you just believe what you read in the catalogues and on the data safety sheets, you won't always get the right answer. So going that extra yard has really made the difference here. So with that, with those final observations, I'll thank everybody for joining and close there. Thanks very much.